Good morning, everyone. I want to thank Dr. Hong Do Lee from the Ingle Lab at the FM Kirby Neurobiology Center, and Dr. Seung-Wha Hu of the Marcia Hagas Lab here at Harvard Medical School for inviting me to kick off what I know is going to be truly an extraordinary day. The fact that we get together like this every year is worth celebrating, because today in this room, we have some of the most important minds working in some of the most essential fields that will define the future of health and well-being of every one of us. Over the course of the day, you're going to hear from leaders in genetics, molecular engineering, computational biology, neurobiology, hematopoiesis, bioengineers and energy homeostasis, and artificial intelligence. As exciting as that agenda is, nothing today means more to me than the chance to stand here and speak with the Chinese students and scholars of Harvard Medical School. The last few years, it has not been an easy time to be a Harvard student, and it's even more anxious time to be an international student. At Boston Children's, we are deeply committed to the students and scholars who have come here from around the world to learn from us. This is a core belief, one that's foundational to who we are. We've been the primary pediatric teaching hospital for Harvard Medical School since 1903. That's something we're proud of. That partnership has been the source of world-changing advances in pediatric medicine for generation after generation. We are all here today because we are part of that. We want to build a better world, a healthier world. And even though the word children is right there in our name, so much of the work we do together, work that will be led by so many of the students and the scholars sitting right here, is of benefit to all of us. Harvard's leadership in that work cannot be overstated, and neither can my appreciation to every student in this audience who has stayed dedicated to this work. Together, Boston Children's and Harvard Medical School form the densest cluster of research activity anywhere in this country. And this symposium, right here, right now, is a perfect example of why that matters so much. Everything you'll hear today makes one think unmistakably clear. Global learning and global collaboration in research, in teaching, and in care are essential to our future. So let me close with this. Thank you. Thank you to everyone who came to share their work today. Thanks to everyone who came here to learn, and to the leadership of Harvard Medical School for bringing us all together today. Thank you.
Thank you, Dr. Churchwell, for the opening remark and the continuous support for our association. We truly appreciate that. So next, Hong Do Lee is going to present a gift on behalf of HMS-CSSA. And I'm going to take a photo as well. Thank you. With that, let's begin our first session on genetics and genomics. So the moderator for this session is Dr. Zheng Sun. Zheng Sun is an assistant professor of medicine at Harvard Medical School and associate scientist at Brigham and Women's Hospital. His research focuses on the human microbiome, host microbiome interactions, and the microbiome-associated chronic disease. So Dr. Sun, please take it from here.
Well, good morning, everyone. I hope you all had a chance to enjoy the breakfast and coffee. Now let's get enjoy for the science. Well, it's my great pleasure to welcome you to section number 1. Thank you. Well, genetics and genomics have transformed not only how we understand biological variation and human disease, but also what we are able to measure, engineer, and ultimately treat. So today we are privileged to have 3 truly distinguished scholars, and without further delay, let's get started. So it's such an honor to introduce our keynote speaker today, Professor George Church. Professor Church is a Robert Winthrop Professor of Genetics at Harvard Medical School, Professor of Health Sciences and Technology at Harvard and MIT, and the founding co-faculty member of the Wyss Institute. Few scientists have shaped modern genomics as broadly as Professor Church. His key contributions include genome sequencing, molecular multiplexing and barcoding, next-generation sequencing, personal genomics, multiplex genome engineering, and synthetic biology. His work has helped move biology from an area focused primarily on reading genomes towards one in which we can increasingly write, recode, and engineer them. He is an elected member of both the National Academy of Sciences and the National Academy of Engineering. So Professor Church will join us virtually today and will speak on genome-wide, transcriptome-wide engineering, how and why. Professor Church, thank you for joining us. And everyone, please join me in welcoming Professor Church. Professor Church, the stage is yours now.
Thank you very much for that kind introduction. Hopefully you can, I'll share my slides and hopefully you can hear and see it. Okay, I can see it, that you can see it. Okay. And I will have hopefully finish a few minutes early so we could have even more time for Q&A. We'll be talking about genome scale and transcriptome-wide engineering, and I'm going to emphasis on why we do this. Too often we do it because, or we seems to be doing it because we can, because it's there. It's, there's a lot of information to be had, especially on the analytics side. I'm going to focus a little bit on how that analytics turns into synthetics and clinical use. Okay, so this is the 18th annual. It's great to be here. These are some of the organizations I'm thankful to, and there'll be individuals that I mention as we go forward. We have some amazing exponentials going on. It's important to appreciate these, that they seem to go slow at the beginning and go fast as they mature. But for example, even as fast as we were going in the early 1980s, if we extrapolated that trend line, it looked like it was going to take 6 decades to get to an affordable human genome, and similar issues with synthesis. Instead of 6 decades, it ended up being more like 9 years. The slope is constantly changing, not plateauing yet, but it could. And this, a lot of this was due to employing multiplexing concepts, where you have shared liquid handling, not necessarily picking and placing a billion things, but doing it all at once. This has resulted in a very high quality as well. So it's high quality is 10 to the minus 10th. And we want to maintain that. So 7 quick topics. A lot of this is themed on accelerated evolution. We do this via libraries, not just artificial intelligence, but libraries to check on the reality and find the best solution. And then we'll talk about recording and recoding different things, and then differentiation, delivery, more on development in the context of de-aging, and then a little bit on speciation. So this is one of the first of our genome scale efforts, and the objective was to get to optimization of a complex biochemical pathway, and we wanted to do it in a very high throughput way. We found that we could generate, this was combinatorial. We didn't specify every single genome, but we made 4 billion genomes per day per one person, Harris Wang, who was a graduate student at the time, now full professor at Columbia. Anyway, we took up to 25,000 cis-regulatory elements, either promoters or protein ribosome binding sites, and would change those in front of each of up to 24, 26 genes. A few of them foreign to the organism. So this is lycopene. It's the red color of many fruits and vegetables, and it's industrially significant. And in 5 days, we got, 3 days, we got 5-fold improvement, which was really quite remarkable back then. This is what we called multiplex assisted genome engineering. We've done many different field biologies here. One of them was Fit Biomics, where we did ENL, we isolated it from athletes, and now this is now a product that's available thanks to Jonathan Scheinman and his team. And then here's another one where we went, in this case, Max Schubert and Braden Tierney, postdocs in my lab, went out off the coast of Sardinia and found a remarkable cyanobacteria that grows faster than the other and aggregates in a very useful way. The cultivarium and the Nili, Ostrov, and Henry Lee have shown that we can get an organism that grows twice as fast as the fastest previous organism, E. coli, and this is being harnessed for biotech, but also many other non-model organisms are being harvested and made to use advanced molecular genetics tools at cultivarium. We can store information, both developmental information and physiological information involving concentrations of compounds, time series, can be encoded in DNA, as Reza Kolhart showed, and others. It was initially developed for encoding a book, but we think of it more usefully as a flight recorder, which records biological information inside of the organism in a tissue-specific manner. So you can access the flight recorder for specific tissues. Jay Shindure and Seth Shipman have also been involved in this, both in our lab and since then. And we estimated we got on the order of sub-terabyte amounts in the first set of mice, and we think that by exploiting the so-called junk DNA, we could get up to an exabyte per mouse stored. Not all of it needs to be read to be useful. We can be very selective about it. And given that we can record physiological information, then optic and auditory senses are basically physiological. They change from biophysics of seeing and hearing into signal transduction. We can harness that into DNA recordings, and so we can make the equivalent of flying camcorders. We also would want to apply these tools, these single molecule tools and evolutionary tools, to electronics. So molecular electronics, molecular transistors, we published a paper on this in 2022, where a large CMOS integrated circuit is peppered with single molecules of variety of sorts that you can bridge the source and the drain for the electrons with a gate, which can be either alpha helix, double helix, or long, thin polymers, and then you can decorate them with single molecules of polymerase or other molecules, and you can detect single nucleotide triphosphate binding. We would like to turn this radically into not just a CMOS circuit, but into something where we can do large libraries, as you'll see again and again in this talk. So that was recording and molecular electronics and metabolism, but the next one is recoding. We want to change the genetic code of the organism and ask, why are we doing this? For every one of these, we're asking, why we do this? And the answer is, we can make biological firewalls, we can non-canonical amino acids, and most importantly, we can make virus resistance. So here's some of the useful amino acids that can be substituted that go beyond the natural 20 or 22. Some of our favorites are fluorogenics. This is like a GFP in one amino acid instead of 238, and it's sensitive to environment, so you can actually use it as a sensor in a way that's been challenging to engineer for GFP. We also have nucleopeptides or nucleobase amino acids, which can be used at the intersection between DNA and protein recognition. Here's some of the data from fluorogenic amino acids. We see fluorescence is dependent on the concentration of the ligand to which the protein binds and causes a different environment for the fluorogenic amino acid. It's time sensitive and ligand concentration sensitive. This is thanks to Erkan Kuru and Jonathan Richier, Alison Flores, and others. And then finally, the third thing that one can do with recoded organisms, the first two were done with one codon change. This one involved 2 codons, or 3, depending on how you look at it. We took 2 of the 6 serine codons and turned them into leucine codons. This was done in such a way that the host organism is using synonymous codons and is protected. It's in on the game, but all of the viruses that come in haven't been recoded, and so they can't adapt to the new code, serine and leucine being chemically quite different. And so essentially every one of these 2 out of 6 serine codons that's present in essentially every protein of every virus is now non-functional leucine. And so you can see on the far right of this data on plaque-forming units per milliliter. We start with very high levels of viral input, including viruses that we found from not just in the lab, right, but also in the wild. We found some farms and lakes and so forth. And this was undetectable replication in these strains of any virus that we've tested. And in theory, that should be essentially impossible to escape. This is thanks to Akos, Svena, and Reagan. It's published in Nature not so long ago. So those are recoding. Now we want to, just like there's a genetic code, there's a cell type differentiation code, which we didn't know a few years ago any bit really much of it at all. And we had some information from Human Cell Atlas. This told us what genes were expressed, but it didn't tell us how to get to that cell type through the complex developmental biology that goes on from the time we're a fertilized egg to the time that we're doing aging and death, all developmental processes. So we wanted to look at these cell atlases, are very complicated data sets, and glean out of them possible recipes for getting differentiation. And we were concerned that we would need to have high levels, that we would have to have very sophisticated timing and amount of the levels of the genes. We were concerned that transcription factors alone may not be it, and so forth. But it turned out, at least for a lot of hundreds of over 300 recipes, we have found that it's actually quite easy. And we'll come back to that in a moment. In addition to the developmental biology, we want to take advantage of the existing diversity of tissues so that we can target particular gene therapies to particular cell types. And so just like there's a code for making the different cell types, there's a code for finding them when, in, say, an intravenous injection, where essentially the blood is taking it to within 100 microns of every cell type. And to do this, we used a combination of machine learning and multiplex libraries, which we call ML. This does use AI a lot to get us close, but then we can get up to 1 million different AI-based designs, protein designs for the capsid that's doing the delivery. And we can take that and inject up to 1 million into even very expensive animal models like macaque, non-human primates, and test 1 million at once. And not only test 1 million viruses at once. We can test lots of different organ types at once from that same animal. So we can get a huge amount of information from one animal, and then we can take that information and feed it back into another round of AI and do even better. And so using that ML, we've found, for example, and this is mostly thanks to Pierce and Eric who were postdocs in my lab and co-founded Dino Therapeutics. We found things like transduction up to 99% of the cortical neurons are, or striatia are expressing the payload, the barcode, and at fairly low dose, and up to 100 times higher brain, or 280 times higher brain transduction, and 50-fold lower liver transduction. So you want to avoid the liver because almost all lipid nanoparticles and viruses tend to go there if you don't do something about it. So here we've done something about it for brain, muscle, eye, and a growing list of different tissues. Okay. So we have, back to making cell types, we have on the order of 2,000 transcription factor open reading frames, and we use them immediately to see what subset of them could act singly as singles. We felt that most of them are going to require complex mixtures, but we would just at least see what the baseline is for singles. Could have been nothing, but we found 290 different cell types, different recipes of singles, and you can see here are single transcription factors. 10 of them that produce endothelial cells, 10 that produce neurons, and 10 that produce fibroblasts. And we all know that there's more than one endothelial type, neuron, and fibroblast, but this was kind of first pass to see what we could get. And this is thanks to Alex and Parastu, who published this paper in Nature Biotech, which really got us going on this. We had done a little bit back as far as 2014, and then started the company GC Therapeutics. This is the 2014 paper where we showed we could get bipolar neurons that could produce action potentials. And this was thanks to Volker Buschkampf and others. And Volker has continued to work on this, and I urge you to take a look at his most recent paper as an independent researcher, where they've made reproducible human neural circuits. To put this in perspective, there are very few reproducible circuits in any naturally occurring animal, with the exception of organisms like C. elegans. But here we're at the dawn of making reproducible human neural circuits with excitatory, inhibitory, and even a variety of non-action potential based neuronal communication. These almost all of the experiments I've been describing here are at one point or another done using resources from the Personal Genome Project. This is providing open source data and cell lines to the community. And in fact, it was chosen by the NIST and FDA as standards for genomics, and now standards for cell biology in the form of stem cells and their differentiated derivatives. Now, Alex and Parastu, I mentioned, have gone on to make, for example, one brain cell type, which is oligodendrocyte precursor cells, which wrap the myelin around axons for rapid communication in, say, your white matter in your spine. And this helps with demyelinating diseases. For example, there are animal models of multiple sclerosis, and these can be cured by injection of these human oligodendrocyte precursor cells. And that is that's the basis of a therapy that's moving its way toward FDA at GC Therapeutics. One that has already made its way into phase 3 clinical trials is called Fertilo. It is we were aiming for at both Gamito and Ovell, two spinoffs two graduate students of mine started as they were leaving, aimed at getting eggs for in vitro fertilization, and discovered that in making the whole ovarian complex of granulosa cells plus egg precursors, we could get granulosa cells to mature them, and so we could use the natural patient cells, egg cells, and mature them, and that resulted in about 3 times lower hormone treatments, which is a huge advantage in terms of pain that a very significant fraction of the women have in these up to 24 hormone injections. So Dina is CEO of Gamito, and this is looking very promising in IVF clinics. Okay, so now de-aging is a special case of development. We have 90% of death is caused by age-related genes and age-related diseases. Therefore, if you had to pick one thing that would have a huge impact on medicine, we think this is it. I've put in bold, out of these 312 genes, I've put in bold some of the ones that we've pushed into gene therapies. So gene therapy is a very good way of getting high specificity if you have a multi-gene family or a multi-isoform, splice isoform, which we have taken advantage of. It's, I think, well established that aging is a lot of different interacting systems, and just fixing one of them might buy you 2 years, but to really get at aging, you have to solve all of them at once. That is likely to involve polypharmacy. Polypharmacy was something the FDA did not encourage decades ago, but now, at least for cancers and infectious diseases, it's very common to have 5 or more drugs at once. And that's what we're aiming for aging. Now, here's an example of a combination therapy, like I said, polypharmacy, with 3 genes. This is much talked about, Yamanaka factors. We abbreviate them OSK for 3 of the 4. And these were developed by Noah Davidson and his team at Rejuvenate Bio. He was a postdoc in my lab who tested out 48 gene therapies at once, and then picked some of the more promising ones. And this was one where when they're injected intravenously at the median life expectancy, so half of the animals are dying of natural causes of old age. So this is very late in life. We don't have to start preventing aging early on for this to have an effect. And here you can see a very significant shift to the right of the blue triple gene therapy OSK relative to the black control set. I think this is the first example of gene therapy for these Yamanaka factors, and probably any gene therapy aimed at with a survival curve. If somebody says they're working on longevity, then they should really have to have a survival curve. If they're working on age-related diseases or just rejuvenation in general, then it's less necessary. Here's another survival curve, in this case a single gene that we discovered, we being Alex Plessa and his team, which is the RNA splicing factor. The OS and K were all transcription factors, as were all the things that I mentioned for differentiation. But this one's RNA splicing factor. You can see a very significant shift to the right on these survival curves when this is delivered. And it wasn't just aging. When we go to the FDA, we're interested in curing diseases. And here's 15 different measures of frailty, including some age-related diseases. And you can see, for example, just evident from the graying and balding on the older controls. Now, we got into this thanks to Pedro de Magalas, who was a postdoc in my lab, who established these age-related databases. And we look for outliers here. Homo sapiens is already an outlier, so this makes it challenging for us to become an even bigger outlier. And nevertheless, we think we finally have the tools to deal with this extreme complexity. We have these exponential tools, which are really paying off. But we published on bowhead whale and naked mole rat. Capuchin monkey is also an outlier, meaning high maximum age as a function of adult mass. And at the other end of the spectrum, very short-lived, unusually off the axis, short-lived are some of the rodents. Okay, so one of the ways of aging that I've mentioned is gene therapy. Another way is replacement of cells or organs. We can replace an old organ with a young one, and it's been shown that things like young blood has an effect. It's not completely understood, but it's very well reproduced in multiple labs. And so we've been exploring organ transplants, not just for the aging component, but also there is a worldwide deficiency in sufficient organs. And even if you had adequate number of human organs, we can make organs that have various advantages. For example, in immunity, maybe we could eliminate the immunosuppressants that are used in human-to-human transplants. We can make pathogens resistance. Every one of these has been demonstrated in animals or cells. DNA damage, cold, dehydration, cancer, senescence, resistance to all of these are possible. And so, for example, here's an organism that can survive, the Siberian salamander can survive -55 centigrade and -35 for many, many days. Most other things that people have talked about actually are not completely frozen and not nearly this temperature. So in terms of immune rejection, we've made, and other safety features of transplantation, xenotransplantation from pigs to humans, we've made 69 germline edits. This includes all the major sugars that cause a very intense and immediate rejection, clotting factors, complement, cascade in the blood as well, and these endogenous retroviruses, which the FDA was very concerned about decades ago and still is, and we decided we would just eliminate those right up front. 59 edits, got rid of them all at once. We've done this in multiple strains of pigs. They don't come back even when the surrogate is producing those viruses. They're resistant to reinfection of those viruses. And this is thanks to Luhan Yang and her colleagues at Agenesis. Luhan was a graduate student and a postdoc on my lab, and then wrote this nice science papers and co-founded Agenesis. So thanks to that. Here's some data on DNA damage that Brad English is working on, and some references to the literature, not from our lab, showing changes in non-homologous end joining, homologous recombination, radiation resistance. These are in human cells, and even in E. coli we can see this. So that's looking very promising. We have xenotransplant clinical trials. We've had 4 patients so far, and we've been approved by the FDA to have another 30 patients, which is moving very quickly now. Tim managed to get to a bridge to human kidney after 271 days, and then Bill and we have more coming along. And finally, just everybody wants to know what the update is on de-extinction. I want to point out that for every extinct organism, there is an endangered one. We're not always looking to bring back an exact copy, although we think that the exponential tools are getting to the point where we could. We're getting very good at reading ancient DNA with high coverage and quality. Anyway, so the ones that are endangered are the red wolf, the Asian elephant, chimpanzee, the dunong, and the numbat. And so here's our 4th example of field biology, the red wolf. And these are partially engineered gray wolves that have been engineered in the direction of dire wolves. thanks to Beth and Ben and others in our organization. This was 7 genes for the woolly mouse and 20 edits and 14 genes for the wolves. So I just want to thank all the people who are involved in this. Here, I mentioned the Personal Genome Project as a source of a lot of our data on multiomics, and even the highest quality genome in the world now, 10 to the minus 10th, telomere to telomere, happens to be one of the PGP volunteers because of the consenting mechanism we have that allows use for commercial use and even re-identification or non-de-identification. Here's an alumni get-together many years ago, and very thankful to the very active current lab and the very active alumni network, many of the CEOs and so forth. That's a full stop, plenty of time for questions. I'll stop sharing. Thank you.
Thank you very much, Professor Church, for that visionary and thought-provoking presentation. Now we have approximately 10 minutes for the questions. Well, maybe I can begin with one. Professor Church, your work has expanded the scale at which genomes can be read and written. So as genome-wide and transcriptome-wide engineering becomes increasingly feasible, so what do you see as the most important remaining bottleneck? Are they from delivery, from biological repeatability or predictability, or safety issue, or our ability to define the appropriate engineering objective?
Well, certainly all of those are true and important. I think that it's going to take multiple exponentials to keep going. The reading and writing, epigenetic reprogramming are all very important to keep those going and building up the database and the AI that allows us to use new combinations, sometimes going far beyond anything that we've observed. So for example, for some of the AAV, I didn't mention, we can make 28 out of 28 amino acid changes, meaning 100% substitution in a key part of the surface that is recognizing the different tissues. So that combination of these tools with AI is incredibly powerful. But it's just another tool. I mean, it's a tool that helps us with our main core molecular tools, like the molecular libraries. And in terms of targets, I think I've already said that I think age-related diseases is the most important one, and maybe tied for that are age diseases of poverty, many of which have been solved, but they need to be properly distributed and made affordable. I mean, for example, inherited diseases is something that probably has 100-fold return on investment, and it's fairly quick return on investment and doesn't require huge governmental input.
Thank you. So my next question is, much of therapeutic genome editing has focused on individual gene or variants. So what conceptual or technological advance will be required before multiplex engineering of like a complex polygenic phenotypes can become predictable and clinically responsible?
Well, it's multiplex engineering is kind of a gradual curve. I mean, it can be very fast, exponential, but it's there's no clear point where less than that's not multiplexing. So, for example, we have Luhan Yang and I at Keyhan have Luhan's team at Keyhan has produced 6 multiplex CAR T therapy that's made it through, I think, about 20 IIT patients, IIT being an amazing program in China. And now that preliminary data has been used and accepted by the FDA for expanding that in a clinical trial in the United States. So that's 6. The xenotransplantation of pigs to humans, that's 69. I think we are getting into the hundreds now at Colossal for non-medical use, but clear phenotype to genotype progress. And I think the next big step will, and probably sooner than anybody expects, we will be doing whole genome synthesis. I described one of them for E. coli, but 1,000 times larger, all the ones we're really interested in next, equal like plants and animals, and even human cells. So I think that there we can make millions of edits all at once and make many different genomes to test them, just as we would make many changes in the capsid protein all at once and make millions of those and test them. So I think that's where we're headed, is full genome synthesis at any scale. And it will surprise some people, just like the arrival of an affordable human genome was surprising to people that had been planning on it being $3 billion.
Professor Serge, thank you very much. So my name is Yin Zheng. So I have a question regarding your, you sliced about 7 areas you are working on. Among those 7 areas, so which one do you think will be have more like a clinical impact in next 5 years, and which one will be more easily to became like the commercial successful? Thank you.
Yeah, it's a little hard to say. I mean, sometimes I'm surprised how fast things go, and sometimes how things that are completely worked out are slow. So, for example, CRISPR was far from completely worked out when we published our first paper in January 2013. But nevertheless, Jennifer and Feng and I and David Liu and Keith Young incorporated Editas, and it just went very quickly. It was just within a couple of months of publishing, 10,000 labs had requested it. So at the other end, and now there's FDA-approved drugs for hemoglobinopathies like sickle cell. At the other end, you have completely worked out next-gen sequencing for diploid genomes that are well understood, and this could save millions of dollars per patient, which is the typical cost of gene therapy. If you just pay a couple $100 for whole genome sequencing at the appropriate points in one's life, like, for example, preconception. And so that's happened very slowly. I mean, we've had those mature protocols for many years, and it's not taking off at the speed of Editas or gene therapy in general, for reasons that aren't entirely well-defined and vary from place to place. One place where it has taken off is in the UAE, which has now got 80% of their citizens sequenced, compared to, say, the United States, where 0.1% of its citizens are sequenced. This is a huge public health opportunity that's being missed in some countries and not others.
Thank you very much. I have one follow-up question. I work in the bioanalysis field. So in the bioanalysis field, in those kind of your new tech, what kind of the new technique you like to see or you like to emerge or maybe in demand in the next few years for characterize the gene editing the field? Thank you.
Yeah, I mean, I think it merits emphasis and even repeating that that whole genome synthesis at the mammalian scale is incredibly important. It's one of these things where we don't realize how much we need it until we've been using it for a few years. And so it's a bit of a chicken and egg, but I'm quite convinced that we will have whole genome synthesis and testing in germline or in somatic cells in the next few years, and we'll be making, just like we're now making, we're sequencing millions of genomes at 6 billion base pairs each. We will be synthesizing millions of genomes at 6 billion base pairs each, and analyzing all the complexities of genome to genotype to phenotype by having millions to test at once, just as we do millions of different AAV viruses. So I think that's, if I had to pick one, that would be my top pick.
Thank you. We can have one final question.
Thank you for the great talk, first of all. So this question is about more like toxicity here, because first when CAR-T came out, everybody think, oh, this is one shot to cure. But 2 years later, there's like a secondary malignancy. And from your point of view, what do you think, the real mechanism and any data you saw that I don't see you want to share with us? Thank you.
Well, CAR-T doesn't quite yet fall into my category of ultimate and general purpose technologies. It only works well on a few blood cancers. So it's not a general purpose technology like next-gen sequencing or editing or delivery. That said, I think it inspires us. It's one of the first uses of multiplex gene therapies. It's now moving from purely cell therapy to also gene therapy, sort of so-called in vivo CAR-T. I think there's lots of room for improvement, but we definitely need prevention of cancer. Is what almost all of the major public health breakthroughs in cancer have been preventative. They've been, avoiding UV, various carcinogens, infectious diseases like human papillomavirus, HPV, and so forth. So we need to pay attention to the preventative as we're trying to extend ideas like CAR-T into solid tumors.
Thank you, Professor Church. It has been a privilege to have you with us today. Thank you so much.
Thank you, and thank you all, everybody.
Okay, so our final presentation brings us from genetic discovery. We'll give the time to fix this small issue. Well, our final presentation brings us from genetic discovery to one of its most compelling clinical realizations, using naturally occurring human variation to reveal regulatory mechanism and create new therapeutic strategies. So it's a great pleasure to introduce Professor Vijay Sankaran. Professor Sankaran is a Jane Allen Paradise MD professor of pediatrics at Harvard Medical School and attending physician at the Dana-Farber Boston Children's Cancer and Blood Disorder Center, and associate member of the Broad Institute and an investigator of the Howard Hughes Medical Institute. His laboratory integrates human genetics, functional genomics, developmental biology, and genome engineering to understand how genetic variation shapes hematopoiesis in health and disease. His work on the developmental regulation of fetal hemoglobin and the BCL11-1 regulatory axis helped establish the biological foundation for therapeutic strategies that have transformed the treatment landscape for sickle cell disease and beta thalassemia, including the first approved CRISPR-based medicine. So his research exemplifies a particularly powerful arc in biomedical science, from human genetic observation to causal mechanism to final the translation. His presentation today is genetic influences on human hematopoiesis. So everyone, please join me in welcoming Professor Sankaran.
Great. Well, thank you all so much. It's such a pleasure to be here and to be able to be part of this fantastic meeting. And I want to thank the organizers for the invitation. So my goal in the next 25 or so minutes is really to talk about some very recent work, but I want to contextualize this in the broader framework of what we've been thinking about and some of the things that were mentioned in the introduction. So each of us is thought to have about 100,000 blood or hematopoietic stem cells that reside within our bone marrow and that are responsible for maintaining our health and immunity throughout our lives. This group of stem cells needs to produce millions of platelets, red blood cells, and a variety of white blood cell subtypes every second to maintain our health. And this occurs through this well understood and extensively studied process of hematopoiesis, which I'm depicting on the left here. Now, this is arguably one of the best understood examples of cell differentiation that we know of in physiology. We understand a great deal about how this process is regulated, how this process occurs both at a descriptive level as well as at a molecular level. And yet, I would argue that as a pediatrician, much of what we observe in our patients and much of what we observe in general in humans cannot be readily mapped onto our understanding of this process. And so we've viewed this not as a limitation, but rather as an opportunity to take advantage of naturally occurring variation to better understand different aspects of hematopoiesis. And as was mentioned in the introduction, our interest in this really started about 20 some odd years ago when at the time, we were interested in trying to understand why the most common monogenic disorders in the world, including sickle cell disease, which I've depicted in this electron micrograph, as well as beta thalassemia, another disorder that's due to mutations in beta hemoglobin, can have such variable clinical manifestations. We knew that there were patients who could be incredibly severely affected, having frequent clinical symptoms, and yet there were other patients who actually would be relatively asymptomatic. It was long known that during human development, there is a switch from a fetal form of hemoglobin shown in green, known as gamma globin, to this adult form of hemoglobin, which is shown in purple, or beta hemoglobin. And this occurs shortly after development, but persistence and persistent production of this gamma globin or fetal hemoglobin molecule was long known for decades to actually be able to ameliorate symptoms in both sickle cell disease and thalassemia. And yet the molecular regulation of this process was not that well understood. And so after lots of failure, we were fortunate to be involved in some of the earliest studies looking at genetic variation between individuals underlying this variation. And this enabled us to identify a number of different loci, including this region on chromosome 2 within the BCL11A gene. And work that we and others were able to do to follow up on this enabled us to show that BCL11A is a key and direct repressor of the gamma globin gene, such that when it's well expressed, which is happening in all of your bone marrow precursors right now, there's suppression of gamma globin and predominant expression of the adult form of hemoglobin. And at the earlier stages of development, there's lower expression at the protein level of BCL11A, and this then enables more robust expression of gamma globin, de-repressing it, and thereby enabling more fetal hemoglobin expression. And this did not just teach us about a fundamental developmental process, but importantly, actually enabled lots of subsequent translational and clinical work that's really enabled a variety of major approaches targeting this axis as a therapeutic approach. And I'll just give you 3 examples of this. There's been work here and elsewhere that's been employing lentiviral silencing of BCL11A, and this has now resulted in clinical effectiveness in a variety of trials and ongoing clinical studies. There's been work to target the enhancer of BCL11A, both here in the US by Vertex Pharmaceuticals and CRISPR Therapeutics, as well as in China by in Shanghai through a number of clinical studies. And this has resulted in the successful approval of Casgevy here in the US and other products that target this axis by really targeting an erythroid enhancer of BCL11A, thereby reducing its expression and inducing fetal hemoglobin. And finally, there's been a number of trials, both from Beam Therapeutics here in the US, as well as from Correct Sequence Therapeutics in China, that use base editors to target the binding sites of BCL11A in the gamma globin promoters. And this has then resulted in de-repression of these genes and clinical effectiveness. And just to give you one example, of course, probably one of the best understood examples is the example of Casgevy or Exocel, where this enhancer of BCL11A is targeted, thereby de-repressing the fetal hemoglobin genes and enabling clinical amelioration in both beta thalassemia and, as illustrated here, in sickle cell disease. And this has shown clear effectiveness where, as you can see here, after infusion of the gene-modified stem cell product, patients actually go on, as shown by the green bars, to actually have significant amelioration, in this case, showing patients having clinical symptoms, in this case, acute painful episodes, as indicated by the blue diamonds, where there's the majority of patients showing amelioration after infusion of the gene-modified stem cell product. I've also had the privilege to meet a number of patients who've received this therapy, including patient one in this particular study, Victoria Gray. And being able to meet Victoria and other patients like her has really been telling, not only for the fact that they report what's seen from the clinical data in terms of the clinical effectiveness, but really this has been transformative in their lives. And in fact, Victoria reports how before she can travel with her 3 children or her husband, she was able to do many things she wasn't able to do before, and was actually spending far less time in the hospital than she was once upon a time. So this has really been an incredible thing to see. Now, given this early success that some of the discoveries we were fortunate to contribute to have had, we've been very motivated to think more broadly about how genetic variation can teach us about this process of hematopoiesis and where there might be therapeutic opportunities. And I would just briefly, in passing, mention that even for very well understood phenotypes like fetal hemoglobin regulation, I think that there's still much more to be understood. And as we're expanding genetic studies, such as this work led by Chunji Guo in our lab, we've been able to identify additional loci. And critically, those additional genetic variants that we've identified have taught us about new biology. For example, the role of activation of gamma globin through this NRF2 BAC2 axis that we're identifying. And this has given us, I think, a more refined view of how this process is occurring and might even suggest new therapeutic avenues by which we can pursue fetal hemoglobin induction. But what I want to talk about today in the last part of this talk is really to give you a sense of where we are going and work that's ongoing, where we think we can not only apply this to maybe more traditional hematopoietic disorders, but really start to think more broadly about this. And so, as I emphasized here, there's been a lot of work in thinking about the monogenic disorders that affect the hematopoietic system, sickle cell disease, thalassemia, a number of bone marrow failure disorders, and immunodeficiencies. And these are being targeted actively by both ex vivo gene therapy and gene editing approaches, as was mentioned. There are a number of companies and academic groups moving this towards in vivo therapies, and that would be an amazing advance for so many of our patients. But assuming this is all successful, and I think that there's a clear path by which this can be successfully applied, could we actually start to apply some of the principles of understanding genetic variation and use that to identify targets for other diseases that are not traditional hematopoietic diseases, but where there are contributions of the hematopoietic system? And I've just given you a few examples, but I really want to focus on one particular example today of how we might be able to think about preventing thrombosis. Now, cerebrovascular and cardiovascular ischemic diseases are estimated to affect about 40 million individuals worldwide every year. And the current approach to treating individuals who've had these arterial thrombotic events is typically to try to prevent secondary events by using oral medications like aspirin and Plavix that prevent secondary events from occurring, that inhibit platelet function. And so this has, of course, been of great benefit, but we know that compliance can be an issue. And even when patients are appropriately compliant with these medications, that there can be patients who continue to show high platelet reactivity, even in the setting of appropriate therapy, and who continue to have secondary events as a result. And this is shown here in this clinical study that's illustrating these patients who continue to show resistance to many of the existing therapies. And so this has made us wonder, are there more effective avenues to actually think about one-time therapies that could prevent platelet aggregation and yet allow for the possibility of reversal? And how might we do that? Well, as I started off with, of course, our focus is really on how we can manipulate the hematopoietic system and how we can use genetic insights to gain into this process. And so we wondered, could we target hematopoietic stem cells and thereby prevent the downstream progeny platelets that are produced from actually aggregating? But how could we do this in a reversible manner? And recent work from Jonathan Weissman, Angelo Lombardo, and a number of other groups have enabled the development of epigenome editing approaches. And these are approaches where CRISPR-Cas9 will be directed to a particular genomic loci, but rather than modifying the genome directly, these actually recruit different epigenetic modifying enzymes. And most recently, this has been enabled the introduction of DNA methylation at specific regions of the genome through the introduction either of DNA methyltransferases like DNMT3A, or by recruitment of endogenous DNA methyltransferases through the use of histone tails, which have been enacted through this CHARM approach. I would just contrast this with other epigenome editing approaches like CRISPR interference that are transient in that they modify histones but should not be propagated to daughter cells. And so this caught the attention of Chen-Yi Ye in our lab, a postdoctoral fellow, and she wondered, could she apply this approach in hematopoietic cells? Now, as I mentioned, in theory, what should be happening is that this de novo methylation that's introduced upon recruitment of these DNA methylation enzymes like DNMT3 to specific loci should introduce cytosine methylation at the targeted locus, and that this should be then by maintenance methyltransferases propagated across cell divisions, particularly DNMT1. So this should then be propagated faithfully across different divisions as cells are both being maintained as well as they are differentiating. The challenge here in applying this in the hematopoietic system, though, is that as I started off in the beginning, hematopoietic stem cells can produce a range of different cell types and also need to self-renew rarely, typically about once or twice a year in our bone marrow to be maintained. And we know that during these processes, there's dynamic changes in DNA methylation as shown by Peggy Goodell, Christoph Bach, and others, and that there can be dramatic changes with hundreds to thousands of loci having altered DNA methylation during differentiation, either towards the myeloid or lymphoid lineages. So this makes it a challenge to think about applying these kinds of approaches in this system. So would this work? So initially, Chen-Yi went on to actually try to apply this approach and introduce this approach using mRNA delivery into primary human hematopoietic stem and progenitor cells. And then ask the question, if we target a convenient surface antigen, in this case CD47, which you can readily detect and which is expressed across all hematopoietic cell types, could we actually silence it in a stable manner, at least in vitro? And indeed, what she could show in comparison to CRISPR interference, where we're transiently suppressing expression over a few days, but then this is actually no longer suppressed as subsequently, is that either by introducing DNA methylation with CRISPR off, or with this CHARM approach, we could stably silence CD47 in the setting of a couple of weeks in culture of these hematopoietic stem cells, both within the broader hematopoietic stem cell compartment, as well as within the more finely phenotyped population of hematopoietic stem cell enriched populations marked by CD90, as shown here. Importantly, this silencing of CD47 expression was accompanied by robust methylation at the promoter of CD47, as shown here using bisulfite sequencing, and importantly was also accompanied by suppression of mRNA production of CD47, as I'm showing you in the plot on the right. In addition, we could take these hematopoietic stem and progenitor cells and differentiate them towards different lineages in vitro. So for instance, we could take these modified hematopoietic stem cells and differentiate them towards the erythroid or megakaryocytic lineages. And even in those settings where CD47 retains expression, we could sustain the silencing that was seen of CD47 in the setting, really suggesting that even during differentiation, this approach could be preserved. But we wanted to apply this in a clinically relevant setting. And so as one of our first targets, we chose to target the glycoprotein 2B3A axis. This is therapeutically targeted by a number of antibodies currently that prevent thrombosis and is a key target where a variety of assays have been worked out to actually prevent platelet aggregation. And so we wondered, could we apply this kind of approach to actually prevent platelet aggregation in this setting? So our approach is rather simple here. We would introduce charm into hematopoietic stem cells. Then from those stem cells, either in vitro produce platelets, or as I'll show you later, we could also show that this is sustained in vivo as well, and then attempt to examine whether this would prevent platelet aggregation in a variety of settings. So as you can see here, by introducing charm and silencing ITGB3, one component of the GP2B3A complex, we could effectively silence ITGB3 at the level of proteins within megakaryocytes or within differentiated platelets. Importantly, if we take differentiated megakaryocytes and examine global gene expression by RNA sequencing, we can see that the ITGB3 was really the only gene whose expression was impacted in this setting. And in fact, in hematopoietic stem cells, there was no genes whose expression was affected. So despite introducing methylation at this region, we could see that this was relatively limited to this single target. If we look across the genome and examine whole genome bisulfite sequencing, we can see that once again, the methylation changes are really only limited to the single locus that we've targeted using charm. And really, we did not see major changes in other regions in this data that was analyzed by Wanying Xu in our lab. And critically, this region of methylation that we've introduced is limited to a 1.2 kb region within the promoter of ITGB3. So there's really no methylation that's spreading beyond this or to other regions. We then could go on, and with Kelly Maklis and Maria Barachina at Children's in the Vascular Biology program, we could examine both properties of the megakaryocytes that were produced as well as the proplatelets. And importantly, morphologically, we could see normal megakaryocytic morphology, normal patterns of the multilobulated nuclei, et cetera. But we could see selective silencing of ITGB3, which also encodes CD61. And importantly, if we allow these cells to differentiate further and produce platelets, these so-called proplatelets, we could see normal morphology of the proplatelets that are produced, normal function, but with the absence of ITGB3. So other than the absence of ITGB3, we're having normal platelet production, normal megakaryocytes, and this can be quantified here as shown on the right side. We then wanted to look functionally, how is this impacting platelet aggregation? And so we could mix these modified culture-derived platelets or CDPs with healthy donor platelets and then attempt to examine, if we label these with two different colors, what is the degree of aggregation. And while normally with either ADP plus a thrombin agonist, we can see appropriate aggregation, or in the setting of this thromboxane agonist with ADP, we can see appropriate aggregation. This is entirely suppressed by the introduction of a charm and targeting ITGB3. We also could see that using a variety of assays, that if we stimulate with these agonists, that platelet aggregation here as assessed by expression of P-selectin is entirely normal in this setting. So the platelets are functioning and activated normally. However, they are not showing activation as assessed by PAC1, an antibody that binds to the active conformation of GP2B3A. And importantly, they're not binding to fibrinogen as shown by this fluorescent fibrinogen molecule that we can show. So really suggesting that we're specifically inhibiting an aspect of platelet aggregation and function through this targeting. Importantly, we can also show that the methylation is maintained during self-renewal, in this case, in serially replating colonies that are formed from different progenitors in the hematopoietic system, and the methylation is maintained. And critically, if we perform xenotransplantation, where we take these human hematopoietic stem cells, introduce them into mice, and then wait several months and look at long-term engraftment, we see no difference in engraftment of any hematopoietic lineage that we can measure in the bone marrow of these mice. But importantly, the methylation is maintained. And if we derive megakaryocytes or platelets from those, we can see that there's silencing that's largely maintained of IgGB3. I would also just emphasize that this is reversible. So if we introduce a Cas9 with a TET enzyme, we can actually culture these cells, methylate IgGB3, and then reverse this by demethylating it with this TET enzyme by introducing this into these hematopoietic stem cells. And if we then differentiate those cells into megakaryocytes, we can re-express IgGB3 where it was silenced before. So we can see effective re-expression as a result of this demethylation. So this is reversible in this setting. I'd also just finally emphasize in the last minute or two that this is broadly applicable. So I've shown you deep characterization for one target, but if we target another platelet glycoprotein GB1BB that's mutated in humans with a mild bleeding disorder, we can actually silence GP1BB effectively, and critically, we can prevent platelet aggregation that depends upon ristocetin or activation of von Willebrand factor in this particular setting. We can also silence this phospholipid scramblase ANO6. We can get effective silencing of ANO6 and prevent ionophore-mediated phosphatidylserine exposure on the platelet surface. As a result, as shown on the right side. And finally, we could also target SERPINH1, which has been shown to be the most differentially expressed protein in hibernating bears and immobile humans, and is thought to prevent collagen-mediated platelet aggregation in this setting. And so we could then target this, effectively silence SERPINH1, and once again, prevent collagen-mediated platelet aggregation in this setting. So really suggesting that across a variety of targets, we can see effective prevention of platelet aggregation in this setting. So what I've tried to suggest in this brief final vignette is really how, as we think about the reach of hematopoietic therapies and the opportunities that we have, especially those that can be really inspired by human genetics, there might be many more opportunities. And I've only had a chance to tell you a little bit about this one vignette, but I think that there's a lot of avenues we're excited about taking in the coming years. And so with that, I hope I've given you some sense of how we at least believe that human genetics can inform our understanding of this process, even for a well-understood example like hematopoiesis. And with that, I just want to thank the people involved, particularly Tian Yi, who's led this work, a number of other lab members, some collaborators who've helped our funding sources. And I want to thank you all very much for your attention. Thank you.
Well, thank you so much for this great talk. Now we have approximately 5 minutes for the questions.
Hello, right here. Thank you so much for the wonderful talk. I have a question that I might have missed, which is about, so it's great to hear all the experimental work, but I kind of lost the connection between how you guys use the human genetic pieces. So so at the beginning of the talk, you show a lot of GWAS plots, and it seems to me that like platelet aggregation is sort of like a polygenic trait. Mm-hmm. And then but I didn't fully connect how do you guys narrow it down to that IgTB3 target, and then to do all the subsequent successful work with it. So
no, that's a terrific question. You know, how did we nominate any of the targets that we went after, including IgGB3? So these are all targets that are nominated from rare human genetic disorders where individuals have some propensity to bleed, but are actually having prevention of platelet aggregation. That really formed the basis of this. I think one question we often get related to this is, what about the risk of bleeding? Isn't this an unsafe approach? And I think that in principle, that's certainly a worry that we have. And I think as we're doing further human genetics, I think that there's opportunities to identify things that actually can modulate bleeding or targets that could modulate bleeding, but that are actually pretty well tolerated, in human populations without severe bleeding, but that can actually prevent thrombosis. And so there's lots of work we're trying to do to actually, more finely identify this. I think in this particular case, we've really taken those targets that we know from human genetic validation, show bleeding phenotypes in humans, but actually can prevent thrombosis.
Great. Thank you so much.
Great question. Thank you.
Yeah, thanks for the wonderful topic about the hematopoiesis, and it can be applied in clinic. So for the sickle cell disease that you mentioned previously, I know there's the genetic modification has been applied in clinic quite well. And from your perspective, like a physician, based on Mass General here and also China, is there any differences for the application? What's the limitation in different areas?
Yeah, so just to repeat, I think the question was really, what are the limitations of these kind of approaches? And I think probably one of the biggest limitations I would say right now in applying these approaches, while we have great therapies, is the fact that in order to apply these therapies, we need to collect numerous stem cells. And in fact, many of these patients, even in the successful phase 3 approved, the phase 3 trial of Casgevy that led to approval, required between 1 and 6 mobilization cycles just to collect enough stem cells. So this means that they were spending between 3 and 18 days to get this like 21st century therapy with CRISPR and genome editing. So on the one hand, you heard George say, well, CRISPR has already been applied, but it's awfully laborious. Like I've been on service and these patients are sitting in the hospital for 3 days just to collect enough stem cells. So I think that's one barrier, is how do we get more, how do we expand more stem cells, how do we better manipulate these cells? And then I think the second barrier I would say is that right now these therapies require patients to undergo myeloablative conditioning. So that means we have to give them essentially chemotherapy to wipe out their hematopoietic system, to suppress their immune system. That's pretty toxic and actually result in a lot of side effects. So I think if we can overcome those barriers, and there's hints that there's ways to overcome that, whether with antibody-mediated conditioning or other approaches, or even in vivo targeting approaches, that I think a key barrier that would enable these therapies to be more broadly adopted would be overcome.
We can have a final question.
And hey, thank you for the talk. I appreciate that. So I have a question here. It's not like an academic. It's more like from insurance company, especially like a Chinese insurance company. They kind of promote. I get bothered often. It's like, oh, we're gonna collect your stem cell. You pay like $5,000. We collect your stem cell. We save it somewhere in the bank, and it could be very useful for the future. And just watching your presentation about whether sickle cell disease, you use stem cell, do genetic modification, and my question for you is more like, is there any like a broad spectrum application for like just collecting stem cell and keep it in the bank?
Well, I mean, it's a really great question. There's been sort of the proliferation many years ago of cord blood banking, I think largely, which is sort of been an area that's maybe become less prominent. I think what's clear is, if a child or even an adult gets a leukemia, we'll almost never use their own stem cells, right? We'll use another donor's stem cells. So I think it's very rare that somebody's own stem cells will need to be used, and usually we can collect those stem cells in most instances, with a few exceptions. So I think in general, I would say there's not really much need for private banking, but I do think that having cord blood banks where cord blood is banked broadly are important. I think with the question that was asked before, with this idea that we have to collect, even for these emerging gene editing approaches, so many stem cells, so many days in the hospital to collect these stem cells. If we could also apply stem cell expansion even to allogeneic products like cord blood, that would be a huge advance and enable many, many more patients to have appropriate donors where currently they do not. So I actually am a believer that if we could expand these approaches, not just for autologous products, but also for allogeneic products like cord blood, we could actually do a whole lot better than what we're doing today. Because many times as a clinician, if a patient has leukemia, they need a transplant, we'll say, well, we have to find an appropriately matched product, but it has to come from an adult where there's sufficient numbers of stem cells. So I think there's a lot to do there.
Thank you, Professor Sankaran. Will, I'm going to use my privilege to ask one more question. Please allow me. So, just in your view, what features make BCL11A axis so special, so tractable, and how those principles could, guide the selection of other genetic modifiers for future therapeutic development?
Yeah, I think it's a really terrific question. Like, what makes BCL11A as the axis of BCL11A so tractable? I mean, I think part of it is there's a fair bit of selectivity if you appropriately target it. So if you target BCL11A everywhere, of course, there's side effects, which is why the approaches either use erythroid-specific delivery of a lentivirus or sort of a lentivirus that's delivered but only expressed in the erythroid lineage, or if you're targeting the erythroid-specific enhancer, or if you're targeting specific elements of the gamma globin genes that are bound by BCL11A. I think that those are certainly the most successful approach that have been applied for cellular therapies. But I would actually argue that there's still a lot more to do. I mean, I think that there's lots of groups trying to target BCL11A using other means, other methods, right? Because these ex vivo cell therapy approaches can only reach a certain number of patients. And so that's where I tend to believe that the more we understand about this axis and the more targets that can be, potential targets that can be defined, maybe the more chances we have to find additional opportunities to target this axis. Because certainly there's currently not a small molecule that targets BCL11A. There's not something that can be broadly applied to the millions of patients around the world who have sickle cell disease or who have thalassemia. So I think that hopefully there can be more advances in a similar way.
Thank you so much.